Hi, my name is Patryk Dabek. I'm a senior in high school, and today I'm going to be talking about my research project dealing with cancer metabolism. So as we know, current cancer therapies are really good at killing cancer cells. They have to be. But unfortunately, they're also really good at killing normal cells. And while doctors try to balance these types of therapies to maintain patient health, unfortunately, many patients with underlying conditions struggle with these therapies and often end up in unfortunate situations. Volunteering with my town's ambulance corps, I've too often seen the struggles that patients have after these therapies. They're forced to call 911 due to healthcare emergencies emerging from the combination of such harsh chemotherapeutics and their own underlying conditions. After learning about cellular metabolism in my biology classes, I continued to read about cancer's metabolism and how its reprogramming made it so proliferative. Through the Warburg effect, macromolecular intermediaries are shuttled into biosynthesis pathways that allow cancer to grow and reproduce, which makes it so dangerous. After all, everyone has to eat. But the more I looked into it, the more I saw that cancer's uniqueness was actually a weakness. With its changes, it actually created a vulnerability through a pathway known as ATP synthase. Even though cancer no longer used the electron transport chain, it used glycolysis, it still needed to maintain its mitochondrial membrane potential. Interestingly, it needed to use the reverse action of ATP synthase to hydrolyze ATP, thus maintaining its membrane potential. And since this hydrolyzation action is not used in normal cells, it had great potential in my mind to act as a selective target for cancer therapy, hypothesizing that if I could collapse this mitochondrial membrane potential, I could destabilize these dangerous cancer cells and inhibit their proliferation. So, to really research this, I looked at it from multiple approaches. To understand if this therapy could work, I first had to find a therapeutic drug that could actually selectively inhibit ATP hydrolase. Oddly enough, looking through pharmaceutical databases, I found that this drug had already been invented. It was used during myocardial ischemia to prevent cardiac cells from killing themselves due to high ATP hydrolyzation. This was exactly what I needed. Utilizing this drug in vitro experimentation in my HepG2 cell line, I was actually able to show that it did destabilize cancer cells and change their metabolism. Foremost, it changed the pathways by which biosynthesis was possible, therefore inhibiting proliferation. Furthermore, mitochondrial membrane potential was in fact destabilized. This depolarization led to a flow of pro-apoptotic proteins from the mitochondria into the cytosol, causing cell death. Now this was very significant, but I think most importantly, it showed that there was potential for this drug to be used to help patients like the ones I've seen in the ambulance have better lives, have better prognoses, and live better, fuller lives. I'm really proud of what I've done with this project, but also so thankful for the recognition that I've gotten. Being able to meet young scientists just like myself at the National Junior Science and Humanities Symposium. Being able to talk with healthcare professionals through the Regeneron Science Talent Search and think of ideas I could never have imagined myself, it really opened up a whole new world to me. That being said, I really hope that my project could serve as an example for the potential of selective cancer treatments, possibly helping to develop a new class of drugs that target the hallmark of cancer, its metabolic reprogramming, to selectively target it, making sure that patients no longer have to deal with the harsh reality that comes along with chemotherapeutics, thus allowing patients to live better and fuller lives.